but we know that species have been going extinct for a very long time. So some people have said, why should we worry about a few more species going extinct now? Well, here's why. Remember this 6 mass extinction graph I just showed you? Well, if we put on that graph a line showing the background rate of extinction, these are the species that we would have expected to go extinct just according to natural processes with nothing major going on. That's only a tiny, tiny fraction of the amount of extinction that's really occurring. And so what we've got here is something that's 100 or even 1,000 times more than what would be expected according to the background rate. So to say that extinction is a natural process and that we should just get used to it and accept it is a bit like saying, well, I'm okay going down 1 or 2 steps at a time, so it should be all right jumping off the top of a tall building. It's definitely not the same, and especially not when you're talking about the consequences for whole ecosystems, which are very complicated indeed. So what is extinction? Well, I brought up here as an example, Lonely George. He was, when he was alive, the last of his species, the Pinter giant tortoise. And because he's now died, of course, that brought about the extinction of the species. But while he was still alive, there were quite a few attempts to try and save the species, or at least save some of the genetic material that Lonely George had inside of him. And so there were attempts to find a mate for George. And in fact, there was even rewards of $10,000 offered for anyone able to find a suitable mate. None of these attempts failed. There were some eggs laid, but they were not viable. And what this shows is one of the key principles of what defines a species, the idea that individuals within the species can reproduce with each other, but they can't reproduce successfully with other species. That is why Lonely George was lonely and why his species is now extinct. There was no other individual to breed with from his species. An extinction, of course, is the loss of a species. So why is that a tricky concept to get our heads around? It all seems fairly simple so far, but unfortunately, like many things in biology, it's not as simple as it seems at first sight. So, for example, these Ensatina salamanders are an example of a ring species. Now, what that means is that although they're recognized as different species, here are all of their names, they can interbreed with each other successfully, but only up to a point. So this species here can interbreed fine with this species, and that can interbreed fine with this species. So there is a kind of chain of ability to interbreed. But at some point, that chain breaks. So across here, they can't interbreed. And so that already brings us into a problem of what we call a species. Do we insist that if 2 things can interbreed, then they must be of the same species? Because if we do, then all of these must be the same because there's a chain of interbreeding possible between them. Or do we insist that if things can't interbreed, that they are not the same species, in which case we must put a break in here somewhere because interbreeding is not possible across this barrier. So that's one of the problems. Another problem is, what about species that don't breed at all? Now, of course, there are species like bacteria, and there we've got a huge problem. But even if we look among the animals, there are examples of this. Like, for example, the Deloid rotifers. This is one here. This is actually an animal, a multi-celled organism, and they've got extraordinary little wheels here, which is why they're sometimes called the wheel animals, that they don't actually rotate like wheels, but they appear to in movies, and they drag food into their mouths. But what is remarkable about this species is that they are all female. Males have never been seen, and they reproduce asexually. So they are only able to lay eggs that hatch out into clones of themselves. That should be a big disadvantage because it doesn't enable them to evolve very quickly around changing environments. And yet, despite that disadvantage, they've evolved into over 400 different species. Coming back to whether or not we count that as a species, we have a bit of a problem, because if the definition of species is ability to interbreed, no individual can interbreed with any other individual. And so every single individual rotifer animal that you see, the deloid rotifers that is, they should count as different species, which can't be true either. And so these are just a few of the problems we've got with defining species and therefore defining and thinking about extinction. How can we have a conversation about something and try to argue to stop it when we can't even define it? Moving on, there are some other concerning challenges around deciding what to do about extinction. And this is quite an interesting one. This is the Californian condor. And in many ways, it's a great success story, because back in the 1987, their population size was down in the 20s. And the decision was made to bring all the remaining living California condors from the wild into captivity for a captive breeding program to save the species. And the program succeeded due to much effort. And here's quite a nice example. This is not actually a parent condor feeding the chick. That is a person's hand inside a glove that's been made to look like the head of a condor in order to make the chick feel as comfortable as possible, ready for release back into the wild. This has all succeeded, and there are now condors in multiple states, and there's around 100 adult individuals in the wild. So that is a success and an extinction averted for the California condor. But not such a success if you are a California condor louse, because when they brought the California condors into captivity, they of course treated them for all diseases and parasites and problems that they might have in order to maximize the chance of a successful program. And that meant making extinct the California condor louse, which lives only on the condor. So ask yourself, are you happy about that? I can just imagine many people saying, well, maybe we don't care about the extinction of this species, but then you have taken a stance about extinction and you've accepted that some extinctions are okay and may even be preferable to bring about. Another problematic issue is within species diversity. So this is the domestic dog, which is rather a special case because all of this diversity is created through selective breeding. It's created by us. But I'm using it as an example that all of these individual dogs here, however different they appear to our eyes, do actually count as one species. And in fact, the wild grey wolf is also a member of that species and can interbreed with dogs. And some people even own wolf dogs as pets. So these are all one species. And yet it would seem like a loss of diversity if we lost all of these different breeds, or certainly if we lost the one wild variety, which would be the grey wolf. So moving on into other territory, here's a question for the audience to ponder over. Which forest would you prefer to take a walk in? Here's one, and I've represented it with a triangle here, and here's another with a circle. So what I'd like to do is just ask the audience to put hands up if you prefer to walk in the triangle forest and then in the circle. So let's do the triangle first. I'm not actually sure quite how to do that, how to see, but hands up if you prefer the triangle forest. Don't see very many hands up here. Okay, who prefers these? Oh, there are a few. Okay, 1, 2, 3, 4, 5. Yep, it's about 6, 7. Yep, okay. Now, hands up if you prefer the circle forest. Okay, so some prefer the triangle, some prefer the circle. Well, that's interesting. So what you may have seen is that the big difference between the triangle and the circle is, in the case of the circle, you have got 4 different species, clearly. But in the case of the triangle, you have more evenly spread distributions of individuals belonging to those species. So you can sort of see with your eyes, really, that this triangle case forest looks a lot more diverse somehow than the circle one, even though it contains fewer species. So as to which of these we prefer, well, fortunately, there's a formula to take care of this. Now, I'm not going to go into too much detail with formulas during this talk, but I'll just show you quickly what we can do with this. We can look at these 2 forests, we can count how many of each species we've got in each forest, and we can represent that as a proportion, so that the proportions add up to 1. Or you could think of that as a probability that if you pick a random tree, that you get 1 from each of these individual species. So these are the values of P1, P2, and P3. And all you do is plug them into this formula, sum them up, apply this math, and you get an answer, a measure of diversity, which is known as a Hill number. But there is also this parameter Q. And you can see that if you set Q to 0, we get back a count of species. So the circle, we get 4 species, and the triangle, we get 3. If we set Q to 2, however, it's reversed. Now the triangle forest comes out as being more diverse and the circle less. So nobody was right, actually, with your voting a moment ago. It depends on your perspective. And this formula rationalizes that and gives us a way to think about it, but it doesn't answer that for us, because it still requires us to put in our perspective about this as a parameter Q in order to give an answer. So here's the take-home messages from the first part of my talk. We want to stop something that's difficult to define, and that for some could be a problem. Certainly it's not something we should overlook. Also, extinction isn't the only bad thing that can happen. We can be left with lots of species, but they might not be the right species, or they might be so rare that for all intents and purposes, they barely exist. And we can't save everything like Lonely George. So we'll have to make some tough choices moving forwards. Which brings me to the Noah's Ark problem. This phrase was coined by Weitzman back in 1998. And the idea was that in the context of limited resources to work on conservation problems, and with species already going extinct, how can we choose which species to save from extinction? How can we choose to use the biblical story that this problem is named after, which animals to put on our ark of conservation here? So there's a few principles we could apply. But before we start talking about that, maybe it's worth thinking about how big the menu is of species from which we can choose. So, how many species are there on Earth? Well, we don't really know, because most species we expect have not been described. That is, they're not known to science. So, well, simpler question, how many species on Earth are known to science? Should have an answer here. Well, we don't really know that either, unfortunately, because there's not a single centralized place that acknowledges unique names of species. And although there are many attempts and many great resources out there, for instance, the Open Tree of Life is one of them, they are fraught with difficulties where the same species has been named multiple times by different people, or where someone's spelling mistake from times gone by has come through and appears like it's another species. So all of those issues are very real. But what I want to demonstrate to you now is the first piece of my own actual research that I want to show this afternoon, which is a way of exploring this diversity of life, this complete tree of life. So I'm going to stop sharing my PowerPoint slides and take you to instead a website that I've been working on really for more than a decade now, not alone, as a team with some other people, particularly Yan Wong, who's been amazing for this project. And actually, Yan Wong, I met him at Leeds, and that's where we first talked about these kind of things. So this is the tree of life. Every leaf represents a species, and the branches show how they're connected through evolution. Now, this might look quite unassuming so far, but there's quite a lot in here to explore. The problem is choosing where to look. So I was wondering what to do for this, but then I thought I'll type in the word target and see what has the name target. So there's several species that have the name target as their common name. Let's try the target beetle. So what we're doing here is zooming in on the target beetle on the tree of life, just as you might zoom into a particular place from the whole globe in Google Earth. And by going on this journey, we can really see just how diverse the tree of life is. It's very easy to say, oh, well, there's 2 million species, but to actually really immerse yourself in that and see what does 2 million really feel like, that's something else. And that's what we're aiming for with this project, to provide a way to explore that. Let's do something very predictable now and look ourselves up. So we are not so closely related to the beetle. So let's just zoom out a little way. You'll see all of the beetles and other insects disappearing into the distance. As we're zooming out, just going back to a broader and broader context on life and on the taxonomy, on the different groups of, in this case, animals, but the plants are also there elsewhere on the tree, of course. And now we've zoomed into the vertebrate and very quickly taken a trip into human. We weren't sure how to pick a single image to properly and fairly describe the diversity of humans, but in the end had the idea of this spacesuit, because we have no idea which individual is inside there, but we definitely know that it's a human. And it also shows something special about what our species has done. You'll also notice as we look at our close relatives that they're colored different colors. So for instance, these ones in the dark red are critically endangered according to the IUCN Red List, whereas these lighter red ones are endangered or vulnerable, but they're still considered to be under threat of extinction. And of course, all of these are our close relatives. So if we zoom out, you can see that really all of our closest relatives, all of the gibbons and the great apes, they're all under threat of extinction. We're the only one that's not. These numbers here tell us about the dates of common ancestry. So we can see, for instance, that it's 18 million years ago during the Neogene period that we shared a common ancestor with these other species. They're our more distant cousins. And as we zoom out, we start to see our more distant cousins still. And we start to see some green leaves creeping in. These are the leaves of species that are not at this time under threat of extinction. And so they're categorized, for instance, as least concern, according to the IUCN Red List. In case you want to read more about any of these species, all you need to do is click on it, and there's a whole load of further information there. So anyway, that's introducing the diversity of life and also the concept of common ancestry and the tree of life, which is something to which we will return in a moment. But let's go back to the slides. So one of the nice byproducts of this One Zoom Tree of Life Explorer I've just been showing you is that we have used that tree as a way to get a popularity index for life on Earth. So this might be an interesting candidate to start talking about if we are going to select species, because it gives us a score of our cultural interest or popularity in every species of life known to science. How did we get those scores and what do they show? Well, we went on to Wikidata, which gives us lots of information from Wikipedia. Particularly, it gives us the length of every Wikipedia page in the English language we used, and also the number of page views over the last 2 years. So if you multiply the page length by the page views, that gives you some kind of an idea about how much interest there is in that particular page. Now, of course, species exist as pages on Wikipedia, and we think that's quite a good way of understanding how much interest there is in those species. Maybe better than Google search, because in Google search, we of course might be looking for, if we type in Apple, we might not be looking for species of apple, we might be looking for Apple computers. If we type in Jaguar, we might be looking at cars, and so on and so forth. Whereas if you go to Wikipedia and you type in those words, it will take you straight to a page that helps you decide what thing it is about Jaguar that you want to look at. And so these page views genuinely seem to be about the species. So we multiply them together to get a value of a page. But of course, a species has more than one page associated with it. So if we take the jaguar, for example, it will have a page for its species, but there will also be a page for big cats in general, and there'll be a page for cats in general, and there'll be a page for mammals in general. And all of those, in some sense, contribute towards the popularity of a jaguar. So we sum all of those up and divide by the log of the number of pages. Why do we do it like that? Well, we kind of made it up, to be honest, but we wanted to find something that was not as strong as just summing them up, but not as weak as dividing by the number of pages and taking an average. So what were the results then? So here we're going to see the top 10 mammals. Polar bear, number 10. Brown bear. Then a range of other great apes, gorillas and orangutans specifically. The chimpanzee and the bonobo, our closest relatives. And then in second place, we have the grey wolf, which of course includes the domestic dog, and all the pages associated. We like ourselves the most. But even though this, I hope, is a reasonable first attempt at a popularity index for all life, there are some issues with it, maybe about it being an index of popularity. So for example, let's have a look at the Asian tiger mosquito, the most popular insect. Now, I don't think anyone would say that the tiger mosquito is the most popular insect, but it is the insect perhaps that receives the most attention and the most interest, not because we like it, but because we hate it, because it spreads yellow fever and dengue, among other diseases. And therefore, there's a lot of interest in this species, at least. So let's have a look at some other ways in which we can think about choosing species to go on our metaphorical arc to be saved from anthropogenically caused extinction. And here we have the aardvark, which is an amazingly fascinating species. It is what we call a keystone species. That is, that it engineers its environment and makes key changes to it that other species rely on to survive. For instance, the burrows that it digs are used by snakes and they're used by wild dogs. What is more, ever wondered why an aardvark has teeth, given that it eats mostly ants and termites? Well, it has teeth, unlike an anteater, because it supplements its diet with something else. The aardvark cucumber. This is the only image I could get of it, but it's evolved specifically to be eaten by aardvarks and to provide them with water in the hot, dry deserts. These fruits, they're related to the cucumbers that we eat, but actually, once the flowers are pollinated, they turn around and burrow under the ground like a peanut does, and then they grow and mature as fruits under the ground, so that the aardvark alone can dig and get them out and eat them, and therefore spread the seeds to other places where it's been digging around. So there's another species that relies on the aardvark. But on top of that, I mean, just look at it. You can see it's got a very unusual collection of traits and features about it. So this is what we could call irreplaceability. And that might be one feature that we could use in order to determine what we put on our metaphorical arc. But all that I've said just now sounds a bit like special pleading. Firstly, we needed a lot of research in order to know all of these interactions about the aardvark, and most species we don't have that knowledge about. And secondly, it sounds a bit subjective and a bit verbal, right? There should be some way to pull out the aardvark in a quantitative way that's backed by calculations. This brings me to the prioritization part of the lecture. And it brings me specifically to a key concept called phylogenetic diversity, which has been around a while, but it uses the idea of the tree of life. So you've seen already what that is. And here we've got a few species connected in a simple diagram showing their ancestry. This is time. So you can see, for instance, that A and B share an ancestor 1 million years ago. That's what these numbers on the branches mean. So here's a question for you to consider and to vote on again. Which of these two environments would you prefer to protect? The one that has species A, B, and C together, or the one that has species D and F together? Okay, let's see hands up for A, B, and C. Anybody? Hands up for A, B, and C. Okay, 1, 2, couple of people, right? 3. Okay, thank you for that. Can you now vote if you would rather save D and F? Lots of people. Okay, so you're already probably thinking along the same lines here. Thanks very much for that. You can put your hands down. So let's talk about this. Here we have, of course, the problem that A, B, and C have 3 species, which seems better. And in a way, there's less extinction if we save these 3 than if we save these 2. But these 2 might capture more unique features than these 3 do. So phylogenetic diversity as a concept is attempting to measure that. Let's have a look. The total phylogenetic diversity of all of these species together is 17.5. How do we get that? Well, we sum up all of these numbers, all of these branch lengths together. 1, 2, 3, 4, 5, 6, plus another, all of these, you get 17.5. What about the phylogenetic diversity of A, B, and C together? Well, we sum up only those branches that are required to connect these 3 species on the tree of life. That's called the minimum spanning set. And in this case, that's 5 million years. Now let's have a look at D and F together. Although it's 2 species, the number of million of years of evolution required to connect them is more. Here now we have a total of 10. And so although the species diversity is higher here, the phylogenetic diversity is higher here. Which of those would you rather save? That may well turn out to be subjective, just as it was subjective whether you prefer to have more species but rarer or fewer species but more common. So how can we use this principle then to do some conservation prioritization? Well, one way is to look at what's called the terminal branch length. So if we look at just these end branches of the tree that connect each species to all the rest, this captures in a very genuine sense the idea of irreplaceability, because the phylogenetic diversity of any group of species can only get this branch of 2 million years connecting to C if it includes species C. This branch is not irreplaceable, because you can get this branch either from having A or B in your set, but these terminal branches are examples of irreplaceable diversity captured by the species that connect to them. And if we look at our aardvark, it has a terminal branch length of 81.6 million years. As you can see from this view of the Tree of Life Explorer, That was actually done with a different color scheme to be more accessible, where we've used blue instead of green to highlight the species that are not under threat. But you can see the aardvark standing out there, filling a big space because it has a long terminal branch length in this visualization. And in fact, it has the biggest terminal branch length of any mammal. But as you can see, it's not in red, so it is not under threat of extinction. Which brings us to a bit of a problem maybe with the ark analogy, because in that story, everything we don't take is going to be wiped out. Whereas that's not true with nature. Some species are going to be okay anyway. So maybe a better analogy is that of an art gallery. So imagine being curator of an art gallery and there's a fire started. The gallery is going to burn down. All the people are out. There's no life at stake here. It's just about what paintings to save. Well, there's two things you might take into account. You might take into account whether it's a particularly irreplaceable piece of work that we've already discussed, but you might also take into account which of these paintings are right next to the fire and need to be saved now or not at all. That is the sense of vulnerability. And so together, irreplaceability and vulnerability will be the things that determine the species that we prioritize. So how do we get vulnerability for species? Well, we use the International Union for the Conservation of Nature, the IUCN Red List, which is the canonical data source for information on species and how much at risk of extinction they are. It's been going for many decades and it has well over 100,000 species in it. Though, of course, those species that are in are heavily weighted towards the ones that we know, for example, especially the vertebrates. More on that later. But we can use these categories as an indication of vulnerability. What we might like, though, is something a bit more than that. So, for example, we might like to know the probability of extinction for species in these different categories. There is a way of getting that from a technique called a Markov model. In a Markov model, species exist in this space and they can move around. They might be vulnerable, they might get worse off and become endangered, or they might get better off and become only near threatened. And so they can move around in this space. And every bit of time that passes, they have a probability of moving to another state in that space until eventually, if luck is not on their side, they end up in the extinct state. You can actually parameterize such a model from historic data of species of the IUCN Red List and how they have changed from one category to another. If we do that, and this is the work of a PhD student in my lab that's not submitted yet, we get this as a probability of extinction as a function of time. Of course, the longer you wait, the greater the chances that you will have gone extinct in that time. And you can see critically endangered, vulnerable, and so on, with critically endangered being reasonably likely to go extinct in 100 years. So that's 0.25, which again might seem like a small probability, but when you consider how many species are critically endangered, that's still quite serious. Over what length of time might you base your calculations? That's an open question. That's something to be discussed. But how, having got this measure of vulnerability, maybe it's worth returning to the question of irreplaceability. So here, imagine species A here, and here we have species B, which is critically endangered. So if species B goes extinct, the irreplaceability of A, which used to be this, has now increased because now this other piece of branch that used to not be irreplaceable is irreplaceable because the other backup copy that we used to have has now gone. Taking that into account, we can have the idea of an expected terminal branch length. That's how long will the terminal branch length of this species be at some point in the future if everything else goes extinct with the chance that we think it will. And that then becomes the new measure of irreplaceability. So combining all that together, we have expected terminal branch length times by vulnerability as a probability of extinction. And that can give us a good measure of which species to prioritize. This is actually actions in some form, in the form of EDGE. EDGE meaning evolutionary distinct and globally endangered, but it's following a similar principle. And that concept has been updated by some of the work that you can see I've cited here. Let me give you an example of the kind of species that comes up as a high priority to go on the arc according to these EDGE metrics, the Cuban solenodon. This is the best picture I could get of it that I actually have legal permission to use, which says something about its state, really, because if they were running around everywhere, people would photograph them and there'd be better pictures to use. But what's particularly special about them? Well, one of their key points is that actually, unlike almost every other mammal, they actually have a venom in their saliva that they use to overpower prey that they otherwise couldn't. So that's one example of how irreplaceable and strange they are, as well as being endangered. And here they are on the tree of life too. You can see on this right-hand side, this is the Cuban solenodon, and it's 35.4 million years is its terminal branch length. There's another 35 odd million years of this branch here back to the point where it has a common ancestor with all the other insectivores, including hedgehogs, moles, and various others. But this branch is quite a lot safer because you see that its sister species, its close relative here, is in blue and is not under threat with extinction. But still, this long branch, having only one backup, even if it's safe for the time being, will have contributed to some extent the score of the Cuban solenodon. And the Cuban solenodon is actually being conserved now, thanks to these kind of calculations, not at all done by me, but these calculations done by the Edge of Existence program at the Zoological Society of London, who have funded work on the conservation of the solenodon as a result. How do we apply this concept to space? Well, so far I've talked about saving species, but what about putting down protected areas and prioritizing space? Can we apply the same principles there? Well, yes, we can, because imagine here's our tree again. We've looked at it quite enough already. And now I've drawn on the right-hand side the range of the species. So this little oval shape here in blue shows where species A lives, and this oval in orange shows where species B lives, and they overlap at some point. Now imagine taking the value in this branch of species A and spreading it evenly over the whole space that it occupies, and taking the value of this branch B and spreading it evenly over the space it occupies, and then taking the value of this shared branch and spreading it over the much larger area that they both occupy. This enables us to spread the value of phylogenetic diversity across space and produce a map. In the map I'm about to show you, it goes one step further and it actually adjusts for the value of the space itself. So for instance, if a space is not at all impacted by humans or very minimally impacted, then a lot of the value is placed there. Whereas if a piece of space is strongly impacted by humans, like a city, for example, then not so much of the value can really be considered to be there. In a sense, it is not as much of an area in the eyes of nature as it would have been if we hadn't impacted it. And if we do all of that for all of the tetrapods, that's the amphibians, mammals, reptiles, and birds together, then we get this map for the globe. It's called HYPE. That comes from human impacted phylogenetic endemism. Phylogenetic comes because it's the phylogenetic tree, and the endemism because it's about the areas in which these species are endemic and their value of the tree is being spread out over, and human impacted because it goes beyond the previous idea of phylogenetic endemism to take into account which parts of the globe have been more impacted by humans. And here we can see key priorities for phylogenetic diversity but only of tetrapods. And we can see well-known areas that are often talked about in the context of conservation, including Madagascar and parts of Central America. One final point from this section is how little we know. So you can see here, for example, if we go to the vascular plants on the Tree of Life, almost all of the leaves are grey. Just a handful are in red or green. Being in grey means that they haven't been assessed by the IUCN, or there wasn't enough data, and so they couldn't really say whether it was endangered or not. So actually, we're in a position where even of the species that are described, almost all of them, we don't know how endangered they are. And so in our prioritization, in our understanding about extinction, can we just rely on our specialist knowledge around the species that have been assessed, or do we need to have some other approaches? That is where the final part of the talk will come in. Just quickly, some take-home messages from this, though. Biodiversity is big, but there are tools for handling that, and those tools are necessary in order to get a grip on extinction. I've shown about how vulnerability, as measured by the IUCN Red List and derivatives of it, and irreplaceability, which could be measured among other ways by looking at the Tree of Life, and factors such as terminal branch length, enable us to prioritize species to place on our arc or to focus conservation attention to. However, a lot is still not known about vulnerability. And it's with that in mind that we come to the final section of the talk in the last few minutes, which is about habitat loss and fragmentation. Now, it's well known that the loss of habitat is a large cause of extinction, or certainly a contributing cause towards the threat of many species. So what do we do if the area that's being lost or destroyed is full of species that we don't know how endangered they are, we know very little about? Well, we have to resort to very different methods to predict how many extinctions we'll get. And so that's the question for this last part of the presentation. What if we take a forest, we destroy parts of it according to a certain pattern, and we ask how many species will we lose as a result of that change? The classic way to approach this problem has been with a species area relationship. Put simply, we take the area of habitat on the x-axis and we look at the number of species on the y-axis. They're both on a logarithmic scale. You put a straight line through them, and therefore you can conclude if you lose this much habitat, then you must lose this many species. But I think that although it's a reasonable starting point, is not the best method to go about solving this once and for all, because for one thing, we don't know from this how that habitat is destroyed. We only know the total amount of habitat that's left. And there have been some debates in the recent literature about habitat fragmentation. That is the process of separating out habitat into smaller pieces. Does habitat fragmentation have a bad effect on biodiversity? Of course, habitat loss has a bad effect, but what about fragmentation? So take a look at these three habitats. Green is habitat, white is human modified habitat, or matrix as it's sometimes called. Now, I'm going to ask you to vote quickly on which of these three you think is better for biodiversity and will involve fewer extinctions. So let's do that quickly now. Any votes coming in? So who thinks the triangle is going to be the best for biodiversity? Okay, 2 people, 3. Any advances on 3? 4, 5? 5. Right, okay, maybe more, 5 or 6. Who thinks the square? Nobody. Who thinks the circle? Okay, 1 or 2 people think the circle. 1, yeah, 2. 2 think the circle. Okay, great. Thanks very much for participating in that. So let's talk about why fragmentation is an issue. There's a bit of a dichotomy here, really, because often when we talk about species diversity, we talk about fragmentation as having brought it about. For example, these tropical pitcher plants, hugely diverse group, many of them carnivorous, many others with interesting interactions with animals and insects, which there's not enough time to go into. But how did they diversify into so many species? Well, when you look at the phylogenetic tree and you look at where they are in altitude, it becomes quite clear that they moved up onto the peaks of mountains and then got fragmented and isolated in different mountain peaks, and each evolved their own way into different species on the different peaks. So fragmentation can, in some sense, lead to increased biodiversity. So why is it so bad then? Well, another example is the Cambrian rainforest collapse. Now, this happened around about the time of former mass extinctions and big geological events. And at this time, large amounts of rainforest that used to cover the globe, but in this study is around the Americas, that rainforest died. And one hypothesis is that that rainforest died, leaving little fragments, and that supported the explosion of tetrapod diversity that was seen following the rainforest collapse. If, however, you put a model into this environment, you can find that you fit the data of what's observed in the fossil record perfectly well, taking into account the fact that what we observe in the fossil record is actually quite a biased view of what was really there. What was really going on, according to the model, is shown in grey. So what this shows is that the model does explain what we see in the fossil record, and it looks like there's a big explosion of tetrapod diversity after the Cambrian rainforest collapse, but actually more likely tetrapod diversity, if anything, decreased, particularly among the amniotes, which are the mammals, reptiles, and birds together. So these are another kind of some of the reasons why habitat fragmentation may have mixed effects. In the case of the pitcher plants, there was no habitat loss. They just moved into different fragments. In the case of the rainforest collapse, there was a lot of habitat loss happening alongside the fragmentation. So how can we get to grips with that? Well, there is a way of modeling it, and there are solutions, but I didn't intend to talk to you about these formulas specifically. I can say that this symbol here represents some really nice math that was done by Stephen Cornell, who Simon mentioned in the introduction. But what we need to know for the purposes of this talk is what is the best case in the short term and what is the worst case in the short term? Well, actually, it's not what anybody voted for. The best case is a splattering all over the place of habitat. And the worst case is one big chunk of habitat. And the reason is that the splattering of habitat all over the place captures more of the diversity that existed in space before. What happens in the long term is also interesting. It turns out that no matter what the structure or what the patterns of loss are, only two things matter. One of them is effective area, how many individuals you have living there, and the other is effective connectivity, which is about how easily individuals can hop around that landscape and disperse through it. So although these two ideas of effective area and effective connectivity around the hopping of individuals through the landscape and measuring how far they can travel on average in their lifetime, these two concepts, although they came out of maths without any preconception of what we would find, they actually meld exactly with what conservationists have been saying for a very long time, that we need to have habitat connectivity and corridors, and that we need to have as much habitat as possible. And for me, that's quite an appealing message about the habitat fragmentation debate. So what is the answer in the long term, at least according to this model, which has its flaws, I must make you aware. Here we have the amount of habitat loss, and here we have the percentage of species that remain. And you can see the best case and the worst case down here. Well, The best case was, again, the random destruction of habitat. The worst case was this artificial one, and the intermediate case is this real pattern of habitat loss. So here's what I think is going on here. All of this arguing about habitat fragmentation is born out of us looking at these patterns with human eyes and thinking, how many pieces are there, or how much edge is there? And actually, nature doesn't see it like that. Nature sees it through its own lens. And so the human pattern identification is not really what people mean by fragmentation. And that's why I think people voted for different squares. Often people do vote for this one here as being best, or some people do. And in a way, that may seem more fragmented to our eyes, but it's also more connected because there's no big gaps that you need to hop over in order to disperse through this habitat. So the take-home messages of the final part of the talk. Fragmentation can lead to speciation, but if it's combined with habitat loss, it's bad. And that also goes, as far as we can tell, for the historical fossil record cases. We've been relying too much on human pattern recognition, in my opinion, to think about habitat fragmentation and habitat loss. And what comes out as, what I think at least is an elegant result, is that these two things that matter are connectivity and area. And that's the end of my presentation. Thank you very much, not only to you for listening and to the organisers, especially for inviting me, but to many other individuals, PhD students, collaborators and funders who've been involved in these various bits of work that I have presented to you today. And as I understand this to be an open conversation, I'm just going to leave us with this slide in case that helps open up interesting things to discuss. Thanks very much, everybody.